My name is Dr. Michael Sandel, and I'm an evolutionary biologist at the University of West Alabama. I'm also a member of an international team of scientists who are committed to understanding the ecology and evolution of Buruli ulcer, which is a devastating and neglected tropical disease. This video is part of a series dedicated to our infectious disease research, and my goal is to describe some open questions about how multiple mycobacteria have evolved into human pathogens. I'll introduce that study system in a minute, but first I would like to acknowledge our financial support from the EEID program, which is jointly funded by the National Science Foundation and the National Institutes of Health. For more details about our project, check out the first videos in this series by Dr. Eric Benbow and Dr. Heather Jordan, and be sure to follow us on social media at EvoPathAmazonia. Before I get into the details of our research, I would like to introduce the concept of the missing link. For a quick example, let's look at an extinct animal known as Archaeopteryx. In 1868, Dr. Thomas Huxley noticed that this creature was very weird because it had both feathers and teeth. He proposed that Archaeopteryx represents a missing link, suggesting that dinosaurs did not actually go extinct, but lived on to eventually give rise to modern birds. Since then, molecular biologists like my colleague Matthew Harris have corroborated Huxley's ideas by looking for ancestral dinosaur genes in the DNA of birds. In 2006, Harris and colleagues reactivated the genes that caused a chicken to grow teeth. So for the first time in about 150 million years, Earth was inhabited by an animal that could develop both feathers and teeth. The story of Archaeopteryx demonstrates the power of integrative research when we derive our questions from the tree of life, or the phylogeny. Today, I'll describe some open questions in infectious disease biology that require the same skills that Huxley developed while he was studying birds and dinosaurs. For the rest of the video, I'll be discussing taxonomic groups of bacteria, starting with the phylum Actinobacteria, and moving into smaller taxonomic groups until we reach the species Mycobacterium ulcerans, which is the focus of our collaborative research. Our team is using the phylogeny to help guide our research questions and to identify the missing links in mycobacterial evolution. The knowledge gaps that we seek to fill are important for public health and biodiversity conservation. The organisms that cause Buruli ulcer and related disorders belong to the phylum and class Actinobacteria. It's not the most diverse bacterial phylum in terms of total species, nor is it the dominant phylum in most microbial surveys. However, members of Actinobacteria are important components of soils and aquatic ecosystems because they synthesize bioactive compounds that structure the microbial communities that they live in. In fact, about 70% of naturally derived antibiotics were isolated from members of this phylum. This is important because, in addition to the bacteria that cause Buruli ulcer, the phylum also contains Mycobacterium Tuberculosis, which is the deadliest human pathogen in history. Our understanding of actinobacterial taxonomy has improved rapidly with the advent of phylogenomics. In 2012, Gao and Gupta inferred the phylogeny for all major groups within the phylum and recovered strongly supported clades containing the order Mycobacteriales and the sister clade Pseudonocardiales. When we compare the morphology of these two groups, we see an important difference in the molecules surrounding the cell, where members of Mycobacteriales are covered by a layer of waxy mycolic acids, shown here in blue. This hydrophobic envelope helps prevent dehydration, but it also offers protection against hydrophilic antibiotics, which partially explains why many mycobacteria rapidly evolve drug resistance. Importantly, the few naturally derived antibiotics that are effective against mycobacteria are synthesized by members of the closely related Pseudonocardiales. So the first missing link in the evolution of Mycobacterial Pathogenesis is the unknown ancestor of the clade containing Mycobacteriales and Pseudonocardiales. What genomic changes explain how these two descendant taxa coexisted and later engaged in chemical warfare? Which group evolved novel weapons first, and were the weapons eventually used against new enemies? We're beginning to be able to address these questions by looking at genomic variation among mycobacterial genera, but we are a long way from understanding the big picture. In 2018, Gao and Gupta found genomic evidence for 5 genera that differ in growth potential and pathogenicity. In this phylogeny, we see that the ancestor of the family was most likely a rapidly growing species with low pathogenicity. As the lineage diversified, clades began to evolve into slowly growing species with increasing virulence. This is paradoxical because one would expect faster growing species to be more virulent than The second missing link deals with the evolutionary trade-off that allows slow-growing mycobacteria to cause communicable infection and disease outbreaks within otherwise healthy human populations. What was the ecological niche of the ancestor to these slow-growing mycobacteria? And what ecological factors selected for the evolution of more virulent life histories? To answer these questions, we are strictly focused on the revised genus Mycobacterium, which is comprised of 4 species complexes, each of which is known to infect humans and other vertebrate animals. 3 of the 4 virulent lineages exhibit a pattern of genome reduction, which suggests that gene loss is responsible for reduced physiological tolerance and specialization to the host microenvironment. This brings us to the third missing link, which involves comparative genomics of the 3 virulent species complexes. To address this question, we need to identify a pattern of gene loss that characterizes ecological specialization. This is important for 2 reasons. First, we need to understand a pathogen's vulnerabilities in order to develop effective medicines. Second, we can use this information to model ecological interactions, which will improve our ability to predict future outbreaks. Among the 3 virulent mycobacterial complexes, the Mycobacterium marinum complex stands out because of its remarkable ecological diversity, from the non-pathogenic Mycobacterium gastri to the highly virulent and drug-resistant Mycobacterium ulcerans. It is this ecological diversity within a closely related species complex that has attracted researchers not simply to find a cure for Buruli ulcer, but also to develop a general model for the evolution of Mycobacteriosis. Our fieldwork is focused on Mycobacterium ulcerans and other members of the marinum species complex found in French Guiana, where outbreaks of Buruli ulcer have been documented for at least 2 decades. One important distinction between Mycobacterium ulcerans and the ancestral lineages within the complex is the presence of a virulence plasmid, which appears to have been acquired through the horizontal transfer from another Actinobacteria. Stenier and colleagues inferred the phylogeny for both the chromosome and plasmid for the Mycobacterium marinum complex, and a takeaway from this study is that our explorations of South and Central America have very high potential for the discovery of the 4th missing link, which is the ancestral plasmid that gave rise to the virulent pathogens of humans and other vertebrate animals. A second takeaway from the Stenier analysis is the lack of mammalian hosts that would be predicted to serve as ecological reservoirs for a human disease. Instead, we see that the closest relatives to Mycobacterium ulcerans were isolated from infected fishes and amphibians. This brings us to the 5th and final missing link, which is the host species that serves as a reservoir for the plasmid-carrying Mycobacteria. Drawing from my previous work on the phylogeny of spiny fin fishes, our collaborative team recognized a non-random pattern of case reports for fish tuberculosis. 2 clades that seem to be predisposed to Mycobacteriosis include the live-bearing piscileids and a clade containing white basses and drums. We will target these taxa while sampling in the field and design laboratory experiments that will help us better understand the cellular interactions between Mycobacterium ulcerans and its various hosts. In summary, our team's past and present research has clarified some important questions that require an understanding of the missing links in mycobacterial evolution. Students and collaborators who would like to join this effort can rest assured that the story is far from complete, and that this research continues to raise more questions than it answers. For more updates, be sure to subscribe to our YouTube channel